Yes, scholars, once again, you are still at TED Talks Education or tedtalks.com. Of course, let's move on with another lesson of biology. Of course, we are still dealing with our topic called genetics, but we have moved very far, and today we can look at multiple alleles. Multiple alleles. We can look at multiple alleles. Multiple alleles. So that is where we are. Now, remember that also this is an exception. Exception to Mendalain is an exception to Mendalain inheritance. An exception to Mendalain inheritance. So now, of course, multiple alleles is a condition whereby a gene, whereby one character is determined by more than two alternative forms or alleles. We say whereby a character is determined by more than two alternative forms, whereby a gene is determined by more than two alternative forms or alleles, of a gene. It's what we call multiple alleles. It is a condition whereby a character or a trait is determined by more than two alternative forms or alleles of a gene. So now, multiple alleles, or the gene for multiple alleles, is autosomal. Multiple alleles, or the gene for multiple alleles is autosomal. Meaning that it is carried on a non-sex chromosome. So, being autosomal, the gene for multiple alleles being autosomal is carried, of course, the gene for multiple alleles is autosomal. So, members, of course, let us first say, before we write this statement, let us first say that an example is ABO blood group system. An example of multiple alleles, or a character which is determined by more than two alternative forms of a gene, is what we call ABO blood group system. Okay? ABO blood group system in humans. in humans. So now we can say that the gene that determine blood groups is autosomal. Which means that it is carried on non-6 chromosome, carried on non-6 chromosomes. Being autosomal means that the gene is carried on non-6 chromosomes. Now, the locus of this gene, where is it located? Where can we find it? Okay? The gene or the locus, the locus for the gene, for blood groups, or for ABO, blood group system. ABO blood group system is isohemaglutinogen. So that is the locus for the gene for blood groups. It is called isohemaglutinogen. We can insert letter A at this position. Isohemaglutinogen, abbreviated as capital I. So now, it means that the genes which determine blood groups are always carried on another gene, or they are found on a locus called isohemaglutinogen. Thus, IA, that is now the allele for blood group A. Allele A, thus allele A, causes synthesis of antigen protein, causes synthesis of antigen protein, synthesis of antigen protein causes synthesis of antigen protein A, on red blood, or say on the surface of red blood cells. Okay? Thus, if it is allele A for blood group A, then of course it will be carried on a locus called isohemaglutinogen, and that's IA causes synthesis of antigen protein A on the surface of red blood cells. And also IB causes synthesis of antigen protein B, antigen protein causes synthesis of antigen protein causes synthesis of antigen protein B. antigen protein. B, on the surface of red blood cells, and IO, okay, IO, we say no synthesis of antigens. For IO, no synthesis of antigens. Now, also say blood groups are determined according to what we call blood factors or antigens within blood. And thus, antigen A determines blood group A. Antigen B, which is a protein, determines blood group B. And antigen O, of course, when we have O, it determines we shall have no antigens. IB, antigen B proteins, and if it is IO, of course, no synthesis of antigens or no antigens. Now, IA and IB are codominant. Meaning that when IA and IB are together, then they can both express in the phenotype. They can both express in the phenotype. So now, IO is recessive. Thus, what happens is that if we have phenotype, or say table showing the possible genotypes and phenotypes. So let us have this table. So I'm gonna have phenotypes, then genotypes. So that is the table. It shows the possible phenotypes and genotypes in reference to blood groups. So if we have genotype IA, okay, then of course, if this is a phenotype, I say blood group A, that is the phenotype. Then the genotype will be IA, or IO. So, if an individual has blood group A, then of course the individual can be homozygous or heterozygous. Then we shall have also blood group B. Here the genotype will be IB, or I carrying B and another I carrying O, which is recessive. We have said that RMO is recessive but carried on a locus or on another gene called I, denoted by I, called isohemaglutinogen. So now, let's proceed. If we have blood group AB, blood group AB, then of course, here, blood group AB. Then here, the genotype will be IA and then IB, because we said that alleles I and B are codominant. When they are present together, they must express in the phenotype to form, they must both express in the phenotype and then form blood group AB. They form a group called AB, determined by genotype IA, IB. And then lastly in the table, another character is blood group O. Blood group O. Here we shall have IO and then IO. That will be the gene. So writing I simply means that the alleles are carried on another gene called isohemagglutinogen. So that is where we are, members. In the next episode, we shall be dealing with an example.